The keynote speaker, will introduce right now. People sit down. Thank you very much. That's Dr. Deanna Barch, who is a clinical from Washington University at St. Louis. And she's a clinical scientist whose research focuses on understanding normative patterns of cognitive function and brain connectivity and mechanisms that lead to in development and rise to the challenges in behavior and cognitions found in illnesses such as schizophrenia and depression. And she does that in the studies by combining psychological, neuroimaging, and computational approaches. She is Vice Dean of Research in the Arts and Sciences at Washington University at St. Louis. She is also the current professor of psychiatry and professor of radiology. And she's deputy editor of Biological Psychiatry, the journal, and editor-in-chief of Biological Psychiatry, Global Science. She is also president of the psychology section of the American Association of Developmental Science. Dr. Barch is on the scientific boards of the Brain and Behavior Research Foundation, the One Mind Foundation, and the Stanley Foundation. She was on the executive committee of the Association of Psychological Science and the Scientific Council of the National Institute of Mental Health. She is a fellow of both the Association of Psychological Science and the ACNP. And she's a member of the Experimental Psychology and a member of the National Academy of Medicine and the American Academy of Arts and Sciences. So, I mean, how many more accolades can one have? Really, superb research. And I thought the work she's doing is really extremely relevant because of its focus on adolescent brain and cognitive development for the exact patient group we are studying, where adolescent and the brain and cognitive development during the period is so important in understanding the development of this illness. So, Dr. Barch, there you go. Thank you so much for coming, and really looking forward.
All right, thank you for that lovely introduction, and thank you for asking me to come today. I really enjoyed learning more about what's going on with schizophrenia. And I'm going to apologize ahead of time if I cough, because I'm recovering from seawater aspiration pneumonia. So just teach you to go snorkeling in the Caribbean and swallow seawater. Apparently that can cause pneumonia. So the work I'm going to talk about today is work that we have been doing as part of a project that I'll tell you more about called the Adolescent Brain and Cognitive Development Study. But I really want to highlight my colleague, Nicole Karcher, who is now an assistant professor in psychiatry at Washington University, who has really been taking the lead on much of our early development and psychosis work. And so I really want to shout out to her at the very beginning, because she's really been a major player / driver in a lot of the work that I'm going to talk about today. I don't have any disclosures of interest. So I know I'm speaking to the converted when, I think we talk about schizophrenia and psychosis as a progressive disease, right? That we know that there are signs and symptoms and things that are occurring prior to the onset of clinically diagnosable symptoms. And, historically, we didn't treat people until they really met full clinical criteria for psychotic disorder. And while some treatments are quite effective, treatment doesn't always work for everyone and for not all components of the illness when it starts after full-blown clinical symptoms have emerged, right? And that's not surprising if you think about psychosis as a neurodevelopmental disorder, because that means the individual has been living for many years with alterations in cognitive function, perception, emotion function, that interact with normative developmental processes and might drive differences in how those processes are developing over time that may be difficult to walk back for some people if not treated until after development of full-blown psychosis. And schizophrenia is, of course, focused on an earlier phase, trying to understand if you can identify and potentially treat people in a more prodromal phase of the illness, either preventing them from, converting into, additional forms of psychosis and hoping to kind of ameliorate the symptoms that they're experiencing even in the prodrome. But what we've been really trying to focus on is, can we go even earlier, right? Could we work more on a very early treatment or even prevention approach to youth at a much younger age who might be on the pathway to developing, kind of psychotic-like experiences or psychotic symptoms that seem in need of treatment. And that's the work that I'm going to focus on today. Now, that is a very challenging set of questions to ask, because we know that, in order to identify kids very early on who might be at risk for developing psychosis, right? If you're just going into the general population, you're going to need a very large sample because the base rate is not high. And there have been many studies over the years looking at familial high-risk studies, but those are challenging in and of themselves. And so the Adolescent Brain and Cognitive Development Study provided a relatively unique opportunity to actually recruit a larger, more population-ish, I'm not going to call it fully population representative sample, that might allow us to start addressing these questions in a unique way. I'll spend just a few slides explaining the ABCD study for anybody who might not be familiar with it. And I do that in part because this is also a completely public data access study. So anybody in this room, your students, your postdocs, can also have access to this data to do whatever kinds of analyses you're interested in. So the ABCD study enrolled approximately 11,874 youth. We started at ages 9 and 10 as sort of the sweet spot between starting early before, for example, kids started to use a lot of substances or started to show a lot of certain forms of mental health challenges, but not so early that we had to wait 30 years to look at outcomes. So we decided on 9 and 10. It was nationwide enrollment, and we are funded to follow them for at least 10 years through to age 21. Fingers crossed that we will be able to continue to follow them after. And we had many goals for the study. Some of it were just understanding normative developmental trajectories, ideally some quote unquote national standards of normal brain development in youth. We're also looking at the roles of genetic and environmental factors, because there is a twin sample embedded in this design. 800 pairs of twins collected across 4 sites. Our site happened to be one of the twin hub sites, as well as blood draws for genetic analyses, and many, many measures of pretty much everything under the sun that you could think about. And originally the study had and it still does have a heavy emphasis on how exposures to different substances might be influencing brain development and behavior and outcomes. But the goals of the study have broadened out far beyond that. For those who are interested, these are the sites of the ABCD. Notably, we do not have great representation of rural communities. That's in part because you needed to be close to a major research center that had access to state-of-the-art imaging facilities to participate in the study. So I think that is a gap in terms of the knowledge base and the population representativeness. But otherwise, we tried to cover as many, parts of the country as possible. It is a pretty extensive battery that we're doing as part of the ABCD. And just to kind of illustrate the design of the study, so we brought people in person at baseline, and we asked people to come for in-person assessments every year. COVID was a challenge, as it was, I'm sure, for you all. And then in between the in-person assessments, we've also been doing phone and online assessments. And so kids and their families come in every year. We actually have already finished year 4. We are now in year 5 and year 6, depending on the age of the kids, and getting ready to gear up to year 7. We cover many different things. So we have assessments of substance use and related factors, youth report, parent report, risk and protective factors, and a variety of biosamples to tap into substance use. And I think this is relevant from the psychosis perspective because of cannabis use in particular. We're trying to get quite detailed assessments of cannabis exposure. Many mental health measures from both parent report and child report, as well as teacher report. Physical health and other related factors, including hormones and saliva and blood for DNA and other health factors. Intensive measures of neurocognition, gender identity, culture and environment, and then perhaps, well, I wouldn't think it's the most important, but every other year we do a full neuroimaging battery, includes structural neuroimaging, resting state diffusion, and task-based analyses. So a pretty extensive battery over time, and all of the data is released annually publicly for people to use. Now, from the very beginning, NIMH invested in this study, and they had two in particular goals for investing in this study. One I won't talk about today, but I could give a whole nother talk about, is suicide and understanding the emergence of suicidal ideation and suicide attempts in young children, but also understanding the emergence of early psychosis risk and how that might interact with factors like cannabis exposure. So we had to figure out a way to start assessing something related to psychosis in 9 and 10-year-old children that would allow us to identify kind of who was starting out with or without experiences, and then what the evolution was of that over time. And I'm sure you might all guess, it's not easy to identify valid measures of these things in 9 and 10-year-old children. Fortunately for us, researchers at UC Davis had already been starting to do this in studies of younger children with 22q11 deletion syndrome. So what they had done was taken a measure that had been reasonably validated in adolescents and young adults, called the Psychosis Questionnaire Brief, and they have been modifying it for use in younger children, including our age range. So we started using a measure, in addition to kind of standard CASADS measures of psychosis, we started using a modified Psychosis Questionnaire Brief, 21 questions that were modified for use in kids. There's a variety of questions. This is just one example. Did you feel like you had special, unusual powers, that you could make things happen by magic? You could know magically what was inside another person's mind. Did you feel confused about because something you experienced didn't seem real or seemed imaginary? Did you feel like other people might want something bad to happen to you? Did you see things that other people could not, or that they did not seem to see? So we started administering this measure at ages 9 and 10. As you might guess or be wondering, at 9 and 10, it's sometimes difficult to distinguish between what might be a psychotic-like experience and what is just typical imaginary normative behavior for those age ranges. At baseline, a lot of the questions, we had an opportunity for kids to respond why they answered that way and what they were talking about, and I read every one of those responses. And it is true that at baseline, some of the kids were talking about, we started this study, I don't know if people remember the movie It with the scary clown. That movie had just come out, and apparently lots of parents let their 9 and 10-year-old kids see It. And so we heard a lot about scary clowns in the closet. But sadly, frankly, there were kids who described experiences that I had heard similar things from adolescents and young adults with emerging psychosis. Now, it's not enough just to know whether kids experience these things. It was also important to understand how distressing they found those. So if a child endorsed one of the items, we then asked whether it bothered them, and if it did, we had them rate how much it bothered them. We did not create the scale. This was a scale that the authors of the measure had already created. So then we could look at not just the presence of these experiences, but how distressing they are. And as I'll show you when I talk about the data that's been accumulating over the years, the level of distress associated with these experiences seems to be a really important indicator of whether they're going to be clinically meaningful and problematic for the children. So we did a lot of work on the original, very first wave of the study, looking at the psychometric properties of the scale, looking at differential item function, looking at whether it, differed across, race or ethnicity or gender, looking at its relationship to, various kind of factors like family history of psychosis. Yes, it was related to that and other things. But now we have been following these kids over time, and what we're really focusing on is understanding distress, duration, and their interaction. And by duration, I mean whether or not these experiences are persisting over time. Because a lot of the literature, if you look at the literature on psychotic-like experiences, there's a fairly high base rate of adolescents and kind of younger children experiencing transient psychotic-like experiences maybe one time, they don't go on to continue to have these experiences. It doesn't seem to be associated with mental health challenges. But when kids continue to have these experiences, and particularly when they're distressing, there's some hints that this may be more of a clinically important set of phenomena that we need to attend to. So one of the things that we've been looking at is, what are the implications of kids finding it distressing and having them last over time? Now, distress is quite straightforward to measure dimensionally. Persistence is less clear to measure dimensionally, right? Because you can't necessarily just sum up scores across multiple assessments, because very different patterns could lead to the same scores, right? So you could have very high scores at one assessment, very low scores at another assessment. If you sum them up, let's say you get a mean of 10, that might be very different than a kid who had a moderate level of distressing psychotic-like experiences at multiple assessments. So we came up with an algorithm where we actually sort of categorize kids as having or not having distressing psychotic-like experiences at each wave. And then we categorize them as persistent if they had them over multiple waves of assessment. And so we created some subgroups here: persistent distressing and transient distressing subgroups, and then folks who had persisting experiences that they did not find distressing, and those who had transient and non-distressing experiences. So 4 groups, recognizing they're somewhat of an artificial cutoff, but we thought this was useful to start to look at this. And then we looked at how baseline characteristics of the sample predicted which kids were going to have either distressing or persistent, or the combination of distressing and persistent psychotic-like experiences. So this is kind of a summary of the overview of the main effects and interactions. So if you look at level of distress, we see that, delays in developmental milestones, alterations in brain volume, specifically reductions in brain volume, impairments in function, the presence of comorbid symptoms like depression and anxiety, disruptions in cognition, were all associated with a greater likelihood of having distressing psychotic-like experiences. On its own, environmental risk factors, social determinants of health, were associated with the persistence of psychotic-like experiences over time. And then the interaction, and I'll show you what those interactions look like. Basically, both persistent and distressing experiences were associated with impairments in function, more comorbid symptoms, worse cognition at baseline, and more social determinants of health factors. And just to give you a sense of what those interactions look like, here's an example for a principal components analysis. We have many cognitive measures. So this is a summary measure of cognition. Higher scores are better cognition. These are kids who have transient experiences versus sustained, right? Red are non-distressing experiences, and blue are distressing experiences. And what you can see, if you look at non-distressing PLEs, even if they're sustained, if they're non-distressing, cognition is doing okay. But if you look at kids who have distressing PLEs, right, particularly if they're sustained over time, they are showing significant impairments in cognition at baseline before we measure this experience over time. As another example, here are comorbid symptoms of depression and anxiety. This graph now looks flipped, right? Because the distressing PLEs, particularly sustained, are more likely to be associated with anxiety and depression at baseline. We also see that with functioning impairment, again, more functional impairment at baseline, problems with school, need for mental health treatment associated with distressing kids who go on to have sustained and distressing psychotic-like experiences. Family history, it's a little complicated. So if you look at distressing PLEs, particularly transient, you have elevated rates of family history. The sustained distressing and non-distressing don't distinguish quite as well in terms of family history. And then here's the environmental adversity measure. So this is like socioeconomic status, neighborhood adversity, exposure to toxins. And again, particularly distressing and sustained PLEs are associated with a greater saturation of these environmental adversities at baseline before they go on to show this trajectory, right? So pointing out to us that looking at both distress and persistence is going to be key to understanding whether this is part of the pathway to emergence of psychotic. Get on my bandwagon. These kids who have sustained and distressing PLEs are very functionally impaired, like in and of themselves. But they may also then be at risk for the development of additional clinically diagnosable psychotic disorders. Okay. Now, you may say, in your experience, right, a lot of kids who show up with clinical high risk syndrome have comorbid depression and anxiety, and sometimes it's not clear what is really about the comorbid depression and anxiety, and what is more due to or related to the psychotic-like experiences. So we did another analysis where we simultaneously modeled psychotic-like experiences, internalizing and externalizing symptoms, and looked at which, baseline factors, again, were more associated with PLEs per se versus these comorbid symptoms. So let me take a minute to illustrate how these graphs work. Okay, so these are different things that could be predictive of mental health experiences. So fluid cognition, crystallized cognition, individual measures of cognition that go into these composites, overall brain volume, cortical volume, subcortical volume, cortical thickness, surface area. These are resting state functional connectivity measures related to the ones that the earlier speaker talked about, the single opercular network, the default mode network, frontal parietal, ventral attention. The size of the circle indicates the magnitude of the effect size of the relationship in a simultaneous regression of PLEs versus externalizing versus internalizing. And this is for the initial level of symptoms at baseline. And then we also modeled trajectories of changes in symptoms over multiple waves. So these are the slopes of changes, right? Blue indicates a negative Z-score, red, a positive Z-score. What you should hopefully see visually popping out at you is basically, for the most part, the magnitude of the effect sizes were much stronger for PLEs than externalizing or internalizing symptoms. And that was particularly true when we were looking at predicting slopes or trajectories over time, right? So just to concretize that, let's look at fluid cognition. So if you, just in order to illustrate these patterns, you could group kids into average fluid cognition, high fluid cognition, low fluid cognition. Here are 3 assessment waves, baseline, year 1, year 2. This is the trajectory of PLEs, the trajectory of externalizing symptoms, the trajectory of internalizing symptoms. And what you can see is for the kids who at baseline had low fluid cognition, they show elevated PLEs at baseline, and if anything, an increasing slope of PLEs over time. Whereas for internalizing and externalizing, they did show some elevation at baseline, but basically they were not increasing over time, right? So greater prediction of PLEs compared to these comorbid internalizing or externalizing symptoms. Here's another category looking at subcortical volume at baseline. So blue is average, purple is high, yellow is low, and you see a similar pattern, right? At baseline, kids who had high levels of distressing PLEs showed reduced subcortical volume. And then they tended to, if anything, show an increase in PLEs over time, whereas we didn't see as much difference as a function of subcortical volume for externalizing or internalizing symptoms. So a little bit of evidence of specificity, or at least stronger relationships to psychotic-like experiences. Now, we also looked more closely at resting state functional connectivity networks, since these have been associated with psychotic-like experiences in older kids and shown disruption in people with schizophrenia in other samples. And we used a cortical parcellation, not quite the same as the one the earlier speaker used, but certainly related, that groups networks into a variety of networks, the default mode network, visual, frontal parietal, dorsal attention. And we focused in particular on the frontal parietal network, the singular opercular network, and the auditory network, although a good reviewer of this paper said, go back and do every single network. So we did go back and do every single network. And if you look at adults with clinical diagnoses of psychosis, including also psychotic bipolar disorder and schizoaffective, this is earlier work one of my students did. What we see in adults with clinically diagnosable psychotic disorders is reductions in functional connectivity among the regions, particularly in the cingulopurcular network, closely related to the salience network, as well as actually overall increased connectivity in the whole brain, which people have argued might reflect kind of a lack of organization into networks. And we've seen this in multiple studies and other people have as well. Got ahead of myself there. And in particular, we see that reductions in the cingulopurcular connectivity is associated with cognitive impairment among adults with psychosis. So the blue line are healthy controls. The other 3 lines are the 3 clinical groups. And in all 3 of the clinical groups, reduced connectivity of the cingulopurcular network is associated with reduced cognition. So this got us interested in wondering whether or not, whether or not we would see something similar. And actually in our psychosis populations, cingulopurcular connectivity actually mediates the relationship between a diagnosis of psychosis and reduced cognitive ability. Okay, so what about in our kids with psychotic-like experiences? Well, we had originally just looked at those few networks, and what we had seen was that the greater experience of distressing PLEs was also associated with reduced connectivity within the cingulopurcular network in these kids. I will note it's not a huge effect size, but it's a large sample. And then when we went back at the request of the reviewer and did every network, this was the only network that showed this relationship, as well as a subcortical network involving the striatum and the cerebellum, which was quite interesting. So again, consistent evidence that these kids with distressing PLEs show functional connectivity differences similar, or I would say somewhat analogous to what we see in adults with psychosis. Now, you noted, I said that the effect size was not huge. We've also been looking at potential methods to optimize associations between brain connectivity and mental health symptoms. And one way to do that is to take a more personalized or individualized approach to identifying cortical networks in the brain. And so typically what we're working with is sort of an average representation of networks in the brain based on averages across groups of individuals. And there's a lot of similarity across individuals in the organization and kind of topography of these networks in the brain. So it makes a lot of sense to start there. But we are increasingly understanding that there are potentially important individual differences in the location and the size of the representation of different networks in the brain. And so folks have been developing methods to try to identify the size and location of cortical networks in individual people's brains and using those metrics to understand functional connectivity instead of what we might call a group average network. So we use an approach called template matching, where you basically use the group average as a template and you identify where in an individual person's brain do you have parts of the brain whose patterns of functional connectivity best match. For example, in red here is the default mode network, and you can identify that representation in each individual. And you can see here's just 4 example participants. That representation is somewhat different across each of those 4 individuals. And so instead of just kind of extracting data from the group average in each individual, we can take their individualized template and we can ask whether we see stronger relationships to mental health symptoms if we take this more individualized approach than the group average approach. So let me explain this. So this is looking at main effects of distress, correlations with we looked at the whole brain, but I'm just for illustration purposes showing you the default mode network, the frontal parietal network, the ventral attention, the salience network, and the cingulopurcular network. So this is the traditional way we analyze the data using group averaged representations. This is identifying networks in the brain that are specific to that individual. And then we call this surface area, but it's basically the size of the representation of that network, right? So more cortex that gets identified as default mode network would give you more surface area of the default mode network. And these are the standardized betas of the relationship between distress and each of these metrics. And what you can see here is that we see stronger relationships, so bigger circles, bigger effect sizes, stronger relationships of distress to individualized representations and also to some extent to surface area than we do to the traditional group average metrics. And if you look at the interaction of distress and persistence, again, where do we see the most disruptions in brain connectivity associated with sustained distressing psychotic-like experiences? Again, we have some evidence that we get stronger relationships to the individualized networks, but even stronger relationships to the kind of the amount of representation in the brain. So for example, here in the salience network, what we see is that those kids who have persistent and distressing PLEs have the least cortex that gets identified as part of the salience network. So a kind of a different way of understanding brain representation. We've also been looking at whether we can identify kids who might be sort of on a trajectory by understanding the similarity of patterns in the brain to those with adults who already have diagnoses of psychosis. So there's an approach that was developed by Peter Kuchinov and colleagues as part of Enigma that they refer to as the Regional Vulnerability Index. This may be something you're looking at in AM schizophrenia as well. And basically what they do is they look at the effect sizes of group differences for whatever illness of interest, right? They've done it for schizophrenia, bipolar, depression, metabolic disorders, Alzheimer's disease, other things. And you can see the pattern of effect sizes. So, here, this is left and right. You can see that, in folks with schizophrenia, they see large reductions in the fusiform gyrus. That's kind of the biggest effect size. And they actually see increases in the superior parietal cortex. So you can think of that as a vector of effect sizes. And then you can do an analysis in a group of individuals where you standardize their data and say, I'm going to correlate the pattern of sort of differences from the average in that sample with this regional vulnerability index. And a higher score would mean that that child has a pattern of brain differences more in the direction of people who have this particular illness, right? So bigger number, more similar to, say, people with schizophrenia. So we just started looking at this in our kids with persistent and distressing PLEs. So 0 would mean there's just 0, no similarity at all. In black, what I'm showing you is the values associated with these regional vulnerability indices for schizophrenia. And then to start to get at specificity, I'm also showing you in purple values for major depression, right? And hopefully what you can see right away is if you look at these kids with persistent and distressing PLEs, they show the largest regional vulnerability indices related to schizophrenia. So the pattern of differences from the average in the sample in those kids is in the direction of being more similar to the meta-analyses that have been done in the patients with schizophrenia and not that similar to depression, for example. We can also look at metabolic disorders as another measure of sensitivity. And again, a little bit here, but not nearly as big as the similarity to schizophrenia. We look at bipolar disorder. Now we start to see, not surprisingly, a little bit of similarity to bipolar disorder, but not, again, as big as we see for schizophrenia. Quite interesting, I actually think, is Alzheimer's disease, which actually is way bigger than the similarity to schizophrenia. We haven't quite wrapped our head around this yet, other than the sort of increasing evidence of, risk for Alzheimer's disease among people with schizophrenia. But among the mental health disorders, we see the largest regional vulnerability indices similar to schizophrenia in these kids who have persistent and distressing psychotic-like experiences. Can someone just tell me time-wise? I don't want to run over. But okay, I'll show you two other quick pieces of data just to hit you over the head. We've also been looking at polygenic risk scores predicting distress, and we're about to start to do the groups of persistent and distressing. I will tell you the data is interesting, but not overwhelming. So these are polygenic risk scores for schizophrenia, educational attainment, PLEs, because there is a PRS score out there for PLEs. There's a cross disorder polygenic risk score, and this is body weight just as a comparison. So we do see that some of these polygenic risk scores are associated with increased distressing PLEs, particularly the cross disorder ones and the educational attainment ones. There's a hint for schizophrenia and PLEs, but not as strong at this point in time as for the cross disorder and the educational attainment ones. But interestingly, oh, and here's CRP, just as another comparison one. What's I think kind of interesting is we were looking at the relationship between this educational attainment polygenic risk score and why it might be related to psychotic-like experiences. And interestingly, what we see is that the mediators of this effect appear to be cortical volume, reduced cortical volume, and impaired cognition, both of which we know are associated with an increased risk of psychotic-like experiences and psychotic disorders. So this may be more of maybe a general risk factor and not necessarily something specifically associated with psychotic-like experiences. All right, there we go. I just want to end with social determinants of health, maybe a little bit strategically, because I think a lot of the work, has focused on the biology and the genetics. Those are not unrelated to the social determinants of health, but the ABCD is also generating data suggesting that psychotic-like experiences are associated with a wide variety of environmental adversities. So we've known for a long time that family SES is associated with an increased likelihood of psychotic-like experiences. So families who are suffering lower socioeconomic status, often, that is associated in this sample and other samples with increased psychotic-like experiences. But we also wanted to look at other kinds of environmental factors. So we looked at factors like neighborhood deprivation based on census data models. We looked at perceptions of neighborhood safety on the part of parents and children. We looked at the number of neighborhood drug offenses, and we looked at lead exposure risk, all of which are independently associated with an increased risk of distressing psychotic-like experiences, even when you control for family SES. So these are add-on predictors. Oh, I'm gonna turn myself off here. And also when you control for parent psychosis, depression, anxiety, externalizing, all of these factors continue to predict. And they seem to be mediated by reductions in cortical gray matter, right? So each of these is associated with reduced cortical gray matter. And if you do mediation models, that mediates the relationship to psychotic-like experiences. So kind of another critical factor that we need to be thinking about. So just to summarize, I think we can identify psychotic-like experiences in children. It seems to be important to look at both distress and persistence. In general, we do see that these are greater in kids who have a family history of psychosis. They are greater in kids who have early motor and speech delays. They are associated with worse IQ and cognitive function at the beginning of the study. We're looking now at whether it's also associated with reductions or failure to show expected developmental increases. They're associated with reduced brain connectivity and networks that we think are important for cognitive control. They are associated with polygenic risk for psychosis and education and cross disorder risk, and with known environmental risk factors. And again, as I said, sustained and distressing associated with the most impairment. Some really key next steps. We've looked over 3 waves. We are about to release data for a 4th wave. So we'll continue to look at who are the kids that continue to have experiences. We're now starting to get to the point where we can look at interactions with substance abuse, because sadly our kids are starting to use substances. So we are seeing, a pretty, I don't have to say big increase, but we are definitely seeing a pretty significant amount of cannabis use now on the part of a number of kids, alcohol use, but definitely cannabis use. The kids in our current wave are now 15, 16, which is an age where kids are starting to do that. And we have a lot of ways of measuring this, including hair samples and saliva samples and self-reports. Kids, I think, have grown to trust us, and they are telling us when they're using cannabis. So we are definitely getting, increased reports of that. We're also interested in linking this to basically the clinical high-risk syndrome. And Nicole, I mentioned Nicole at the beginning, part of her K-23 is to actually do SIPS interviews with a subset of these kids. So she is doing interviews with both caretaker and child for 600 dyads, with the idea that a third of the dyads are kids who have persistent distressing psychotic-like experiences. A third of those who have persistent internalizing symptoms, but not psychotic-like experiences. And then kids who don't have any mental health challenges. We've done 90 dyads so far, and it's blinded, so I can't say anything about what's likely to happen, but I think this will give us some sense of how it connects to the kinds of kids who may be coming in for AMP schizophrenia. Nicole's also funded to look at more data-driven predictors of outcomes using machine learning because we've been doing maybe a more hypothesis-driven approach given known risk factors, but the data set is big enough that I think we can afford to do some data-driven analyses. And ideally, we'd be able to start to identify some interventions earlier on. And if all of this works, we are very much interested in broader screening, and that's something we've been doing with Brandon and others at One Mind is looking at how can we develop more integrated screeners, thinking about 2 stages like Professor McGord talked about. I think it critically interacts with what AMP and other groups are doing though, because screening really is a better thing to do if you actually have treatment resources that you can refer people to. And so identifying treatments and treatment resources and increasing the number of resources available is going to be critical. So I will stop there. I just want to shout out again to Nicole Karcher because she's really been driving a lot of this work. And so I want to really make sure she gets the credit where credit is due. So stop there.
Thank you very much. It was a great talk and beautiful data set that you really used to its maximal advantage. So that is super. Are there any questions or comments? Yes, Salsa.
That's right. The next question, we had a good day everyone. People know that 8% or 8-year-old children have children who are being for and that also happens in people but I really like the approach of interest and secret. But I was wondering if that could be used outside the psychosis world. I mean, there's a big report about communities showing that among all others, they should be in female or material or psychosis. 50% want to happen with a child and adolescent that experience. And the 2 main sources that really drove the high risk to quickly psychotic identification rate was not integrated to psychosis, it was depression and context. So, I was wondering how this expressed persistent model would work outside psychosis. And in that same picture, most people finish quickly criteria of purpose, but they don't have to
Yeah, okay, so I was told to repeat the question. So Council is asking whether we could look at distress and persistence as potential predictors of adult emergence of psychotic disorders for things other than psychotic-like experiences in adolescents. So I think the answer is yes. That's part of the reason why in the diet, Nicole is identifying kids who have persistent internalizing symptoms. I mean, really, if we had uber amounts of funding, we could do lots of different kind of persistent externalizing sense of, persistent depression, persistent. If you could identify kids who might have persistent cognitive challenges in the absence of these other symptoms, I think we could look at, okay, what is that harbinger about, right? Like, is that telling us something about their risk for other kinds of disorders as they can, emerge into adulthood? Or is it kind of, so you can think of it as heterotypic or homotypic continuity, right? Like, there is a lot of evidence for heterotypic continuity where you might have persistent depression early on, and that might not just predict depression later, but the emergence of other things as well. So I think we could do exactly that in these kinds of data sets.
Thank you very much. And yeah, there's a question in the back, please.
Please question about the part of this words, are they being collected in individuals with
Great question. She asked whether we're looking at the polygenic risk scores in folks with Western European ancestry. So we collect data from the entire sample. Those analyses only use the children whose parents reported that, and we also looked at the ancestry scores in the DNA. We looked specifically at the kids with Western European heritage because there is a lot of concern about the validity of applying the polygenic risk scores and other samples, as there are more, the Broad and others are collecting better data sets that we can then apply, because there's probably about 2,000 kids in the sample whose parents identify as Black. There's another, large chunk who identify as Hispanic, so that is a limitation. We can only do those analyses with a subset of the kids.
Thank you.
Thank you, Cheryl.
Yeah, that's a great talk. I had the same. So, polygenic risk score, the effect on persistent stress certainly was mediated by both brain volumes and cognition. You showed that for the psychosocial adversity, that it is brain volumes, but it is the cognition, not a function. So I really tend to work in and the second one is the really interested about neighborhood drug use, because that's a much higher data than some of the others. So I'm trying to wrap my mind around what is actually happening.
Yeah. Okay, great question. So I think you were asking whether a cortical brain volume and cognition mediated both polygenic risk and environmental adversity. And I can tell you, we didn't look at whether cognition mediated environment, so we can do that. That's an interesting question. We haven't done that yet. Neighborhood drug use, yes, I think it's very fascinating. We've been talking a lot about what that might be indexing and thinking about things mechanistically. So we've been, thinking about what is it about some of these like neighborhood factors? How do they impinge on the kid, right? So is it that kids are more likely to be exposed to traumatic experiences if they live in a neighborhood that has a drive-through? Is that indexing? Because it wasn't just general crime rates, it was in particular the number of drug offenses. So we don't know exactly what that's indexing. Like, is it those kids are being, are they more stressed about going out of their home? Are they seeing violence in the streets? Like, is that, something like that that's driving it? So we don't know. Yeah.
On the corner.
Yeah.
And maybe you're feeling at some point that they have been pulled into this it's just happening.
Yeah.
Eric, I think there was one question on Zoom, so I don't know how to access that. So if you can do that. And that's going to be the last question before we close.
Okay. The question is from Judy Thompson. It says, really great work. Any specific relationships based on the type of feeling, unusual thought content versus perceptual alterations / hallucinatory experiences? Absolutely great question. We have started to take a little bit of a look at this. So we've done some like IRT analyses and other things to try to identify if we could shorten the questionnaire, what's the most, could we get it down to 5 items, 6 items? But we haven't really done any sort of systematic analysis of like all of these other things. as a function of individual symptoms, but that's definitely something us or someone else, because it's all publicly available data, could do as well, because that'll be important to know. Because 21 items still takes a while, like it takes our kids 10 or 15 minutes to do this questionnaire, and it's actually one of the longer questionnaires in the mental health battery. And every year, as we aim to shorten the battery, we fight for keeping this questionnaire, as we always say, the NIMH wants it. So, but if we could shorten it, or identify subgroups of items, that would be helpful.
Thank you very much. Thank you very much again, Dr. Barch. It was a great, very inspiring talk and beautiful work. Thank you so much.